I want to go a little bit over some of the stuff we discussed earlier, but concentrate again on specific aspects that I didn't spend enough time on. So let me start with describing the control of epidemics. And remember that the equation that is at the center of the SIR model is an equation that defines how the number of susceptible people, the ds by dt, is proportional times the fraction of the infectious population. And the proportionality constant must involve something called the contact rate, what is the rate at which individuals contact other individuals, and something involving how efficient is that contact in the transmission of disease. So both of these, all of these quantities are hidden inside the term beta. The rest of this is just measures of how many susceptible people are there, how many infectious people are there, and so on. And then the second equation said those infected recover at some rate, and the number recovering is proportional to the number infected. So dr by dt, the number recovering, the change in that in time, is proportional to the number of infected, and the proportionality constant is a gamma. So unit of gamma is 1 over time, so gamma is a rate. So there is a rate that is involved here in the beta, because again this has units 1 over time on the left hand side. So the ratio of these rates is a quantity that is dimensionless, and that, as you know already, is a central point, is a central sort of the core quantity that is defined in the theory of infectious diseases, at least at the level of the simple SIR model. So to stress that again, R0 is a product of the transmissibility of the pathogen, which is a measure of how effective is a single contact in transmitting the infection, the frequency of contact, which is how many contacts are there per unit time, and the time to removal, which is the duration of the infection. So if you know what the number of contacts are per unit time, multiply that by the duration of the infection, you have a measure of the total number of contacts that is made in that particular period. And it's from there that you get this particular quantity R0 equal to beta by gamma, the basic reproductive ratio. But now that you know where each of these parts come from, what R0 consists of, or what are the contributors to R0, this suggests how you might control an epidemic. So really speaking, there are 3 steps in this. The first is to reduce the transmissibility by developing vaccines or using antiretrovirals in the case of AIDS. The second is to decrease new contacts. And this you accomplish by isolation or quarantine through health education programs that say that, look, don't go too close or touch things that people who are infected have touched before you. The third point is to reduce the length of the infectious period. All of these attack either the beta or the gamma term. So this last point here attacks the gamma part of it. And so therapeutics, antibiotic treatments, boosting innate immune response, all of these are ways in which you can reduce the period of time that an individual is infected. So these 3 major points really, all of which come from considering what R0 is made up of, is at the core of health interventions for infectious diseases. So it's not just if you think carefully enough about what goes into R0, and you can do this not just for the simple model that we wrote down, but far more complicated models. You look at the terms that are structured into the definition of R0, you can figure out how to target one or the other of these terms and find out what is the right public health and what sequence of actions can you do to reduce the burden of infectious disease. Okay, so that's one thing which I didn't concentrate on. We've been talking equations for so long, so I thought this class should be a little more about phenomena and controls and so on. We'll do a little bit of equations as well. But let me go back to the idea of metapopulation and describe something that I didn't tell you about earlier, which is how to calculate R0 for a metapopulation. We know how to calculate it for a single simple SIR model. But the idea of the metapopulation is that you have a bunch of different populations, 1, 2, 3, 4. They're connected somewhat weakly. The rate at which they're connected is dependent upon some matrix, the beta ij matrix here. So this is a slight rewriting of the way I wrote it earlier. Remember, I had, I think, X and Y in my earlier description of this model. So I've just gone back to writing S and I instead. This is a term that couples different infecteds in different populations. So this is population 1, population 2, population 3. So there is one term here that refers to that particular population, infections that you get from individuals in that population. But there are also terms in which I is not equal to j. And those are terms in which susceptible people in one population can contract the infection from infected people in another population. Okay? And this is the sort of picture that you should have in your mind whenever we think about metapopulation. We think about fairly tightly knit populations, each describable by a single SIR model between here, 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 but then a weaker link between one population and another population. Typically because people might migrate from one population to the other, people might have social contacts between these populations. There might be commuters who move between population to another population during the day. They go there and they come back. And we wrote down a commuter approximation for that particular result. But here are the equations that you would write down for S and I in terms of a single SIR model. And this is just sort of the picture of the compartments of each separate compartment and the interaction between these compartments. So this arrow here is infected people in this compartment infecting other people in the same compartment. But these longer distance arrows are infected people here infecting susceptible people in another compartment. Okay. So the force of infection in patch I is the weighted sum of the numbers of infectives in the other patches, and we've absorbed the factor of 1 by n into the beta terms, which we can do anyway. So the question is, what sort of definition of R0 covers a multi-group setting? So here you can consider early stages of the infection. The early stages of the infection are described by a linear model in the following sense, that you can define a quantity called, which is defined or termed the next generation matrix, which describes the average number of secondary infections in the ith group that are caused by a single infective individual in the jth group over the infectious period. If I equal to j, that is someone in your group, an infected person in your group, giving a susceptible person a disease also in your group. But you could have an infected person in another group giving the disease to a susceptible person in your group. So that has the structure of a matrix with I and j, where I and j run separately between 1 and n, if n is the number of groups, and k is an n by n matrix. So k encodes the structure of how you acquire infection from other people in the population, from other populations inside the metapopulation. Okay. Once I tell you what this matrix is, you can define the basic reproductive number R0 as the dominant eigenvalue, the one with the greatest modulus of the matrix K. This is also called the maximal eigenvalue. So let me tell you why that's so. So if you have a vector phi naught that gives you the number of infected individuals in each group at the initial time, then if you take K and act it upon phi naught, that gives you phi 1 by construction. Because phi naught is the number of people at time t equal to 0 across the different n subpopulations, the population making up a metapopulation. If I multiply this by K, that gives me the number of secondary infections due to this primary number of infected individuals that is spread out over the population. If I do this again, K squared, K cubed, K to the power 4, if I keep multiplying this matrix, that is at each generation, there is a new set of infections that are created via this matrix K. So at any later point, I can ask how many infections are there in the population, and typically that will give you some vector phi m, which is K to the power m times phi naught. That is the mth generation starting from the beginning. Okay. So now imagine that this K has some set of eigenvectors which we denote by psi j. You can write the initial state phi naught as some sum with coefficient Cj of this. So this is like expanding in your eigenvectors with certain constant coefficients that sit out. Now I can take my K matrix and sort of multiply it m times upon this initial state phi naught. What you will get is that K will act upon the psi j's to give you eigenvectors lambda j's. So what you will finally have is lambda j to the power m psi j and these constant coefficients Cj that sit out here. So this is your phi m. Okay. Now suppose there is a strictly maximal eigenvalue lambda 1, just for choice. Let's assume that the first eigenvalue called lambda 1 is the largest. So I have a sequence of numbers here, lambda 1 to the power m, lambda 2 to the power m, lambda 3 to the power m, etc. It's obvious that the largest one will dominate this particular expansion at long enough time, because that's the one that is growing fastest when I multiply it. So in that particular case, I can approximate this sum by saying that phi m is approximately C1 lambda 1 to the power m times psi 1. Okay, so this is the largest or the maximal eigenvalue. So within this linear description, the number of infectives increase by a factor of lambda in each generation, and this is what is really meant by R0. Okay, so how exactly the infectives are distributed among the groups when n is large is given by the entries of the eigenvector psi 1, where we have said that here psi 1 is this particular quantity that sits here. And the subdominant eigenvalue describes the transient behavior, the approach to that situation. But how long it takes to reach that asymptotic value depends upon the relative sizes of the dominant and the subdominant eigenvalues. That should be obvious for you if you think about it just a little bit. Okay, so let's go back to the main points that I had illustrated about how it's necessary to move beyond the simple SIR model to get an accurate description of disease. So remember the points I said: the SIR model is a deterministic model. There is no stochasticity in it. There is no spatial information in it. That doesn't tell you that, unlike even the metapopulation model, which has some limited amount of space information, in the sense if you think of these as being spatially separated populations, there's at least some saying that, look, if these are weakly connected, maybe they're separated well apart in space. So there is some indirect information about space, but no real information about space in there. This is a coarse-grained approach where we ignore The fact that every individual in a population is different, has different susceptibilities, different rates of recovery from an infection. We just put all of them into these boxes or compartments, the S, the I, and the R compartment. And the final assumption that we make is a mean field assumption, that anyone in the population can contact anyone else. And really the rate at which new infections arise is just proportional to the fraction of infectors within a population. So we talked about this particular point the first time around. So if you remember, we discussed stochastics, we discussed how to take this equation and write it in terms as a sort of master equation with Poisson rates. Then we talked about how to solve that equation using the Gillespie method and describe the Gillespie method as essentially a two-step process, one in which you determine the time until the next event happens, and the second is in which you define which is the event that actually happens. So let's look at the second term in a little detail. We'll come back again to the second point. So in order to do this, this is the sort of simplest generalization that you could possibly think of the SIR model. So first of all, I have dropped the R term because I know that S plus I plus R is a constant. Therefore, R, if I know S and I know what R is. And R only is a sort of box in which I take everything that is leaving the I compartment. It does not affect the S and the I independently. So this is the term that I would write. So look at the changes that have happened. First of all, S is now a function of space and time. So the interpretation of this is the number of susceptible individuals in some small little box surrounding the location X at time t, and this is how it changes in time. So these terms here, this term here, here, are all the standard terms that I would write down if I was writing down an SIR model. All I have done is added, instead of writing just S of t, I have now made it S of x and t. Okay, but each of these x's evolves independently at every point in time because there's nothing to connect different points in x. So if I just stopped here, this would be like having an independent SIR model at each different point in space. Because there is nothing there is x here, x here, x here, x here, x here. But this is not really a spatial model in that sense because it doesn't include, it's just an independent SIR model at every point. Okay. In order to make it spatially sensible, we add what is called diffusion terms on this right hand side. So that says that susceptible people move around. If they initially start here, if you pile them all up here, as time goes on, they will broaden out in their distribution and they move with some, let us say, diffusion constant ds. And similarly, the density of infected individuals also changes with a diffusion constant di. So a diffusive process, remember, is something that spreads out. If I initially concentrate it, the state that it wants to have is one that is flat and constant at a much later time. So these terms essentially provide that. And they're the simplest ways of putting any sort of spatial information into this model. Notice that because this is the del squared term, that couples different points in space. And as you know, if I had sort of switched off these two terms here and solved just the diffusion equation, the solution does change in time and space. Any arbitrary complicated initial distribution of S's will broaden and become flatter in time. And that's really what the diffusion equation, which is this term here, actually does for you. So we can make it a little simpler scale, S of xit plus it equal to 1. Then I have this quantity as di by dt is beta times I, 1 minus I times I minus gamma times I, that's this term here, and the diffusion constant that sits out here. Okay. So this is a simpler model, it's not SIR, this is SI. So then again I have one constraint, which is S plus I equal to 1. So I can get rid of one of these equations, the equation I get rid of is the S equation. I finally have only an I equation which looks like this. And this is where the information about what happened to the S came. S is now 1 minus I. Okay. So this is a simple partial differential equation. It's the first derivative in time and two derivatives in space. It's nonlinear because I and I appear here. There is a minus gamma I term. So if it was just beta times 1 times I minus gamma I, that would be a linear equation. It should be easy to solve. This equation is a nonlinear equation, so somewhat a little harder to actually solve. This equation here is also called the Fisher-Kolmogorov equation in population genetics. And this has interesting properties. It has traveling wave solutions, and I will describe what the meaning and the interpretation of those solutions are. So let's start with this equation and then ask what are the questions that we want to ask. Typically, the question we want to ask might be, if you start at a point and you have one individual at one point, that individual infects other individuals around it, and that infection spreads. Because the whole way we have written these equations says that the infected numbers broaden out if you initially set them up at one point through this term over here. When I say broaden out, how does it broaden out? Does it broaden out like a diffusion front that is moving across, or does it broaden out like a wave that is moving through the population? So that's the question that we want to ask now. if it broadens out like a wave, you expect the following transformation to work. So first of all, you want to look for solutions that are traveling waves that move with speed c. So a good change of coordinate is one that moves along with the wave at speed c. So you write a variable z, which is x minus ct, and change all the variables that appear here. Delta by delta x is delta by delta z, and delta by delta t is minus c times delta by delta z. So I can take the delta by delta x's and delta by delta t's, put them back into this equation, and finally extract all the terms that depend upon delta by delta z and ask what does that equation look like. And the equation, what I want to start with is an initial s equal to s naught everywhere and one little I at one particular point. So if I'm just sort of reinstating the s naught, instead of 1, let it be s naught, and then I'm putting this particular factor in here. This is the original equation. This is the transformed equation here, and now this is a linear equation. This is I times xt. There is delta by delta z here. There is this term here. There is a diffusion term here. And now I can say, okay, this looks a little better because I've now got rid of any initial nonlinearity that existed by making this particular transformation. I can guess at a solution which is just a function of the variable z. Okay, so this is a guess for what I think the solution should look like. Its I of z is some constant here, e to the power r times z. And what I have to fit is this constant here and this constant r. So if I insert it and I sort of ask what the trial solution should correspond to in terms of what are the values of a and r that satisfy this equation, then I get 0 equals di times r squared plus c times r plus beta s naught minus gamma, which is then a quadratic which has to be solved for the value of r. If I do that, I get r equal to minus c by 2 di plus or minus c by 2 di square root of the subject here. Now, this represents for you, this iz equal to this quantity here, represents for you the number of infected. This is the number that must be non-negative, therefore r must be real. If it was oscillatory, it would go above and below a certain mean value, and then there is a chance that that value could be, if you are depending upon the choice of a, that that value could be negative. So that puts a strong constraint on what r can be, and that constraint really says that the discriminant of the quadratic, that is this term here, should be non-negative. If I sort that out, go back here, look at that discriminant again, what this means is that c squared, where c is the wave speed, should be greater than or equal to 4 di times beta s naught minus gamma. So this implies that c is itself greater than the square root of this thing, and this implies that there is a minimum wave speed for waves of infection. Okay, and that form, if you sort out the values of beta s naught, gamma, etc., is twice square root of di times gamma times r naught minus 1. So here you can see that r naught has to be greater than 1. If it was not, then this would be imaginary, because this would be a number that is less than. So the other feature, of course, is that this is a nonlinear model, and a nonlinear model can have multiple different types of waves. In fact, it's not just one wave that is moving out, but the passage of that wave can give rise to secondary waves just behind it. And where these ideas have been applied is a particularly interesting model about the spatial spread of rabies. Okay. So rabies is something that you don't want to get. So if you're ever bitten, if there is the slightest chance that you have been bitten by a dog or a cat that may exhibit symptoms of rabies, please go and get injections immediately. This is a viral infection of the central nervous system. It's transmitted by direct contact. And it's almost 100% fatal if you don't get your rabies vaccination in time. In Europe, foxes carry more than 70% of the disease in Europe, because typically dogs and cats tend to be not feral but already vaccinated against rabies. And so you can think of a single species with subpopulations for this model. They're territorial and the territories don't overlap, so they're sort of separated in space. Rabies induces behavioral changes, so the dogs, they sort of lose their orientation, they move around. So it's good to think that dogs with rabies, that foxes with rabies essentially move diffusively rather than in any directed manner. And this is invariably fatal. So once you contact rabies, the fox tends to die, so that the infected is removed from the system at a constant rate per fox. So the equation that you would write down here is ds by dt equal to minus I times s. This is the second term. This is the diffusion term here. We have scaled various numbers out. Over here, you can take these equations, solve them. So this should be di by dt. I'm sorry, there's a mistake here. You can find a traveling wave with the boundary conditions that s at infinity is 1, s prime at infinity at minus infinity is 0, and there are no infecteds at either ends of the system. And you can study these as early and solve for the wave velocity. So people have done that, and this is one example of what is called the Murray Spatial model, which is related to the model that I showed you earlier. So this shows you the traveling wave at different times, 240, 364, 80, 600, 720 in some units, I think, of days. The Fox density is non-uniform across the country, which means that the wavefront is not circular, because the detailed behavior of the wavefront will depend upon what's the initial condition that you started out with for the Fox density. So this is how, even if you started at time t equal to 0, with one or a small number of rabbit foxes at this particular location here. So this is somewhere in England, used as a model. You can see how, as a function of time, you will spread through the entire country. And in a way, if you trust models like this, you can say that, look, it will take roughly about 960 days for it to reach here, for sort of detectable symptoms for foxes with rabies to be caught and at this particular point here, so far away from the epicenter from where the infection started. So here is a little more detailed model that shows you how the wavefront moves forward. So this is the front of the wave, this is the secondary wave that sits at the back of it, and this is time. So 360 days, 720 days, 180 days, etc. How it moves, how it reconstitutes itself and starts again here. Okay. So these are nice examples because just introducing space into the SIR model, putting it into a geographical structuring like this, and starting with some initial assumptions for how the foxes are distributed in this location, which you can get from standard available data, you can really come up with pictures like this. How does one infected fox at one particular location, maybe someone might have brought it over from the continent where you have cases of rabies, and brought it over to the south of England, actually spreads in time. And you can predict how far it has spread, what is the shape of that spreading. And this is data that you can then compare to various records of public health or veterinary health. Where exactly has the wavefront gone? Should we need to correct the model in any way? Are these time scales correct as far as we know about the infection? And that's really an important point of how we can use this. Okay, so the second part of what I wanted to tell you about, again, as I said, we were getting a little bit more mathematical, but I thought we would go back again because I wanted to combine some level of epidemiology with the mathematical modeling. So I thought I would spend some time today in describing one particular disease in some detail, which turns out to be fairly important. So I want to give you as many details about this disease as possible, tell you a little bit about all of the variables that people consider while modeling this particular disease towards the end. But really what I want to do is to provide you with the phenomena that are known about malaria, a little bit of the history behind malaria epidemiology, a little bit behind what mosquitoes, the nature of the infection, etc., the natural history of the infection of malaria, so that you're on your own, you can think about this particular problem. So much of what I will talk about, at least historically, comes from this nice paper called Ross-Macdonald and a Theory for the Dynamics and Control of Mosquito-Transmitted Pathogens, which appeared a few years ago in PLOS Pathogens. The material on malaria is very easy to find. Sort of standard resource is the CDC malaria website over here, as well as the Scientists Against Malaria website here. So if you do want more information to check that what I've told you is correct, you can go back to these references and check. Okay. So mosquitoes are vectors, that is, they transmit diseases, and they transmit a whole bunch of pathogens causing malaria, filariasis, dengue, yellow fever, West Nile fever, and several others. The idea that mosquitoes could transmit disease, could act as vectors, actually is an old idea. It goes back to someone called Patrick Manson, who showed that mosquitoes transmit a blood-borne pathogen. It goes back to 1877. A gentleman called Charles Laveran observed malaria parasites under a light microscope. So you could actually see that there was the causative agent for the infection was a particular parasite, which he could see under a microscope. The real study of malaria epidemiology can be considered to have started with Ronald Ross in 1897, who showed that mosquitoes transmit those parasites. And then a little later, Reed, Finlay, and Carroll showed that mosquitoes transmit yellow fever virus in studies in Cuba. Bancroft showed in Australia that mosquitoes transmit the dengue virus. Again, this is fairly old, this is 1906. And that's a whole bunch of people who've contributed to this initial understanding of what mosquitoes do in the transmission of disease. This is from the 2014 malaria report. This is a measure of the confirmed malaria cases per population of 1,000. So this is more than 50, between 1 and 50, 0 to 1, and no malaria at all. So just looking at this, you can see immediately that the burden of disease really is in Africa, because in all of these areas here, what is called sub-Saharan Africa, because the Sahara is over here, this is sub-Saharan Africa, And you can see the darkest shades of red are correspond to these regions here, the Horn of Africa from here, and sort of Central Africa at this point. There's a somewhat lighter shade for India, somewhat lighter shade, similar shade for China, etc. This area here, which I guess is Thailand, shows you a sort of fairly dark. Thailand, Malaysia, probably going down. Is that correct? Yeah. Okay. Anyway, we can clarify that later. This is this is Indonesia over here, and South America, especially in the northern parts of South America. All of these are areas that are influenced where malaria plays an important part. In 2015, there were 91 countries with ongoing malaria transmission, and nearly half of the world's population is currently at risk from malaria. But most cases and most deaths really come from Africa, which is why Africa has been a place where the Gates Foundation and many large-scale WHO efforts in restricting the spread of malaria have concentrated on Africa. Between 2010 and 2015, malaria incidence, which is the number of new cases reported in a particular year, fell by 21% globally. So certainly the efforts at controlling malaria were working, and the mortality rates from malaria also fell by about 29% globally and by about 35% among children under 5. As you can see, as you saw from that picture, sub-Saharan Africa carries a disproportionately high burden of malaria, and it is home to about 90% of the malaria cases and 92% of malaria deaths. So malaria, it's mosquito-borne, it's febrile, which means it leads to a fever. Symptoms typically develop within between about 10 days and 3 to 4 weeks following infection. Here are the common symptoms of malaria, just to remind you in case you haven't had it. The sort of standard symptom of malaria is a shaking chill. That typically is periodic, it happens at the same time every day. So you can have a shaking chill where you just shiver. That ranges from moderate to severe. There's a high fever, there's profuse sweating, there's a headache, nausea, vomiting, diarrhea, anemia, muscle pain, convulsions, coma, bloody stools, etc., etc. But the sort of canonical symptoms are really those that come from here: the bad headache, the sweating, the high fever, and the shaking chills. So the WHO estimates most recently, which is December 2016, counted 212 million cases of malaria in 2015 and 429,000 deaths. These are large numbers. What do we know about malaria? So that's what I want to sort of discuss a little further. What we know about malaria, the natural ecology of malaria, involves malarial parasites infecting two types of hosts. One is a human host and the other is a mosquito host. The mosquito host is a female Anopheles mosquito. And this is called a vector because it helps to transmit the disease. All important vector species, mosquito species, typically bite between dusk and dawn. In the middle of the day, you're unlikely to be bitten. Some species prefer the evening, they're called crepuscular, so both sunrise and sunset. Some species bite during the night. The intensity of transmission, that is how effective is a mosquito bite in transmitting a disease to a human, depends on many factors. They depend on the parasite, the vector, the human host, and the environment. So there are approximately 3,500 species of mosquitoes that are grouped into 41 genera. And this is a long history about how the people were just naming things at random, and finally a committee got together and said we must find a more systematic way and more controlled way of assigning a particular species name or a genera name to a mosquito. There is there's a vector research center in Pondicherry, which has a room called the Mosquito Museum. So in the Mosquito Museum, they have records of something like, I think, all of the mosquito species, or a very large number of mosquito species that belong to India specifically. So it's a nice room to see. It's a small room, kept air conditioned, and there's sort of drawers, and you pull out the drawers and you can see sort of stacks and stacks of mosquitoes in tiny little jars. So human malaria is transmitted only by females of the genus Anopheles. Of about 430 Anopheles species, only between 30 and 40 transmit malaria or can be called vectors. This is a distribution of different types of vector, of different types of malaria species. So we are here, we more or less one color here. You can see that there are significant variations. The species here is related to the species here. There is some similarities between different colors. The species that affects Africa is not the species that affects India, interestingly. So if you look at India, the largest numerically is a species called Culicifaces, and there are a bunch of others that are more geographically constrained. There are some particular species that are seen in the northeast. There are some species that are seen in the Gangetic Plain close to the mountains. Okay, so these anophelines, these mosquitoes, go through 4 stages in their life cycle: egg, larva, pupa, and adult. The first 3 stages happen inside water, and they last between 5 and 14 days, depending upon what the species is and what the temperature is. And so it's only in this final stage, the adult stage, when the female Anopheles mosquito acts as a vector. Adult females can live up to a month, but there's a distinction between the wild or in captivity. So in captivity is typically about a month, but in nature, it is probably between about 1 to 2 weeks. So in order to nurture the eggs, the female mosquitoes seek blood, what is called a blood meal, which is the point at which they begin to bite. Each species of Anopheles has its own aquatic habits. Some prefer small, shallow collections of fresh water, such as puddles and hoof prints. You can see these are abundant typically during the rainy season tropical countries. Adult females lay a fairly large number of eggs each time. Eggs are laid directly on water or unique in having floats on either side. They're not resistant to drying. They hatch within about 2 to 3 days. Females are fine with having sugar. So one way to feed mosquitoes is to have a solution of sugar available, but they usually need a blood meal for the development of eggs. Okay, so after the blood meal, the female will rest for a few days while the blood is digested and the eggs are developed. Exactly how long this takes depends upon the temperature, but can take between about 2 or 3 days in tropical conditions. Once the eggs are fully developed, the female lays them and resumes host seeking, and this cycle keeps repeating until the female dies. So the malaria parasite must first undergo 2 stages of development. One is within the mosquito, one is within the human. So there's a first stage of development within the mosquito before they become infectious to humans, and the time required for development in the mosquito is called the extrinsic incubation period. Extrinsic meaning outside the human. That ranges from about 10 to 21 days, depending upon the species and the temperature. How long do these last? There are some estimates for some species. So one particular species called Anopheles gambiae in Tanzania, the daily survivorship ranges from about 0.77 to about 0.84, which means that at the end of the day, if you started off with the number 100, roughly between 77 and 84% will have survived for that day. If you assume that this is constant, this rate at which you lose a fraction of your population every time, this means that less than 10% of these mosquitoes would survive longer than a 14-day extrinsic period. But if you increase that survivorship to 0.9, instead of between 77 and 84, over 20% of mosquitoes would survive longer than a 14-day extrinsic incubation period. So one implication this has is that controlled measures that rely on insecticides, such as spraying inside, actually might impact malaria transmission more through their effect on how long the adult lives, rather than what they do to the population of adult mosquitoes. You tend to transmit more intensely in places where the mosquito lifespan is longer, which means it has more chances of infecting. The parasite has time to complete its development, and there is more time to infect people, and also where it prefers to bite humans rather than other animals. The reason malaria is such a problem in Africa is there are 2 reasons. One is that mosquitoes live longer, so longer lifespan, and they prefer to bite humans. So the strong human biting habit of the African vector species is really the reason why you have more than 90% of cases inside Africa. Transmission is usually seasonal because it depends upon water and peaks during and just after the rainy season. And you tend to have an epidemic whenever climate and other conditions favor transmission in areas where there is very little immunity towards malaria, where it has not entered previously. It also happens when people with low immunity move into areas where you have intense malaria transmission. For example, a refugee is leaving one country and moving to the other. In their original country, they might have had very little exposure to malaria, but in the new country, they would have much more. A little bit about the parasite. So the malaria parasite is a protozoan, it's a single-cell organism. Its genus is Plasmodium, and there are more than 100 species of Plasmodium, and 4 have been recognized early enough to infect humans in nature. So that's Vivax, Falciparum, Ovale, and Malaria. And one more was added to that in 2008, which is Plasmodium knowlesi, which was originally known to infect only monkeys, but then it was realized that it also was an important factor in affecting human beings as well. The most prevalent malaria parasite on the African continent is Falciparum, which is itself responsible for most malaria-related deaths globally. But in sort of outside of sub-Saharan Africa, it's Vivax, which is a dominant malaria parasite. So what happens? What happens when you're bitten by a mosquito? and the mosquito injects a certain parasite into you. So what happens is that in humans, the parasites grow and multiply first in the cells of your liver and then in the red blood cells. Okay, in the blood, there are successively these parasites multiply, they give rise to broods and then destroy these cells, breaking them up. And this breaking up of the cell releases daughter parasites, and those continue the cycle by infecting other red cells. And it's at this stage that you see the symptoms of malaria. So anemia is sort of one consequence of this, as listed earlier. So here's a picture of what happens. This is the infected person here, and the bite of the Anopheles mosquito withdraws a parasite from the infected person. There is a stage of development within the mosquito, then the mosquito bites a person here. It enters the human liver of this particular person. From the liver, it's released into the bloodstream, comes out here, affects red blood cells. They mature within the bloodstream and they finally burst the red blood cell at this point. Okay, so when the mosquito picks up the parasite and during a blood meal, it starts a different cycle of growth and multiplication in the mosquito. In between about 10 to 18 days, the parasites are found in the mosquito's salivary glands. And the name for the parasite at this stage is called a sporozoite. When the Anopheles mosquito takes a blood meal on another human, these are injected along with the mosquito's saliva and they start another human infection. So here is that particular picture. Here is the mosquito biting. This is the release of the sporozoite into the bloodstream of the individual. From there, it moves to the liver, where it enters a stage called the merozoite. In the liver, once it reaches this stage, it infects red blood cells and starts a cycle of continuous breaking up the cell, releasing parasite, and infecting new parasites. And there is a later point where it switches out of the cycle, or some fraction of parasites switches out of the cycle, and creates what are called gametocytes. It's these gametocytes that are absorbed by another mosquito bite, and that reinitiates the cycle of infection within the mosquito. So slightly on the mosquito side, when you draw a blood meal, this is the point at which the gametocytes of the parasite enter the mosquito gut. Over here, it has to adjust to very different surroundings, and this is what makes all of the microbiology of the show extremely interesting, because remember that the environment that the human blood supplies and the environment that the gut of the mosquito supplies are very different environments. So there is a process by which the parasite has to adjust to the insect host of the warm-blooded host, and that initiates a cycle called the sporogonic cycle. Okay, so this is the microgamete becoming a macrogamete, becoming a zygote, reaching the, all of this happens in the midgut, then it leaves here and then reaches the salivary glands. So there's a complex process of microbiology that happens inside the mosquito. And once they fuse in the mosquito gut, they form zygotes. That is the stage here. These develop into oocynocytes that actually move around and burrow into the mosquito midgut wall to develop into oocysts. So this is the oocyst here. This grows and divides and bursts, and so it's after this phase, which is 8 to 15 days, it releases the sporozoite into the salivary gland, and that goes back and when you bite again. Here is the whole complicated cycle both in the mosquito and the human. So as I said, there is a part that happens only in the human, there's a part that happens only in the mosquito, and this sort of keeps going back and forth as the infected mosquito comes, bites a human, starts out the cycle, another mosquito might come, withdraw the particular parasite, and begins all of this back again. There are a large number of quantities that could potentially be important, and so there's the article that I referred to earlier lists a large number of them. In fact, because people have been working on the problem of modeling malaria for at least the last 100, 110 years, everybody has a different notation, everybody has a different way of writing these things. So this is just one way of systematizing people's notations in some common way. So certainly the population density of humans is important in sustaining an infection with malaria. Population density of mosquitoes must be important. The number of infected humans, number of infected but not yet infectious mosquitoes, and the number of infectious mosquitoes. So all of these refer to different stages of the not yet infected but not yet infectious, number of infectious. Then there must be quantities that relate to how mosquitoes and humans interact. So the human blood feeding rate is the proportion of mosquitoes that feed on humans each day. Mosquito survival is important because mosquitoes have to survive long enough to be able to take up the blood meal, to infect, and then another mosquito must be able to come and initiate the cycle within the mosquito. Pathogens vertebrate latent period, that is the number of days from infection to infectiousness in the human, the number of days from infection to infectiousness in the mosquito. the proportion of infected humans that are infectious, or the probability that the mosquito becomes infected after biting an infected human, blah, blah, blah. All of these are sort of large and important numbers. You can also scale them in sensible ways. The prevalence of malaria is the number of infected people divided by the total population here. Fraction of infected but not infectious mosquitoes is Y divided by M. Infectious mosquitoes is the quantity Z divided by M, where all of these quantities were defined in the earlier slide. So Z was the number of infectious mosquitoes, M was the population density of mosquitoes. So dividing Z by M tells you about a fraction of infected mosquitoes. The number of bites per human per day is called the human biting rate. That's defined in terms of some quantity M times A. And there are a bunch of terms here. I mean, don't pay too much attention to them. I just wanted to draw your attention to the fact that when you think about this problem, there are many factors that enter into it. There are factors that just determine what is the density of the host, what is the density of the mosquito, what is the fraction of mosquito bites that are effective in transmitting the information, what is the lifetime of the mosquito, and so on. And all of these appear to greater or lesser extent in any model that you will write down. So what the importance of Ross's work was that Ross showed that if you reduce mosquito numbers below a certain transmission threshold, that was sufficient to counter malaria. This is like our definition of R0 and what we refer to as herd immunity. By reducing the effective R0 to a number which is 1 or less, you ensure that the infection could not propagate within the population. This is something like that. If you can reduce the gross density of mosquitoes which are responsible for infection below a certain amount, that should be sufficient to counter malaria. And what Macdonald did, so these models are called Macdonald-Ross models, to give credit to the people, first to Ross and then to Macdonald later, who modified it in significant ways. He put in latency in the mosquito due to malaria parasite development and said that the survivorship of the adult female mosquito is typically the weakest element in the cycle that I described to you earlier. And so if you could kill the adult female mosquito, or that would be sufficient to reduce its numbers, and thereby vindicate Ross's idea that if you just manage to reduce the number of mosquitoes below a certain threshold, then you could terminate the cycle. Okay, so that was why the WHO invested a lot of money in using DDT as an insecticide to kill mosquitoes in order to eliminate malaria transmission. And then the latency of infection in humans was introduced later by Anderson and May and gave you a separate exposed class in humans. So the Ross-Macdonald model in somewhat modern language really assumes that the number of mosquitoes is constant. Mosquitoes could be either susceptible or infectious. If they're infectious, you call them I. There's a constant human population size, which is H. Humans could be either susceptible or infectious, in which case you would call them Y. You ignore incubation periods. And you say that the rate at which mosquitoes bite humans is proportional to the number of mosquitoes, but independent of the number of people. Okay, just each mosquito wants to bite. The more the mosquitoes are, the more the mosquito bites. Okay. So here is a sort of compartmental model description of what is happening. This is the human set of human compartments and a set of mosquito compartments. So humans can go between being susceptible to being infected, and what affects this rate of infection is the level of mosquitoes. Similarly, infected humans control the rate at which mosquitoes become susceptible, mosquitoes become infected. So that's this greenish line here. So there are these multiple compartments that are connected to each other. But given a compartment model now, and also there is death and birth of mosquitoes that you must account for here, you must also account for the recovery of human individuals there. Okay, if you want, you can put in death of individuals arising from malaria and so on, and you can just make it more complicated. So now with a model like this, you should be able to write down the equations given the experience that you already have. So this is the structure of the Ross-Macdonald model. This is Y dot for infective humans is some constant times I times H minus Y by H. So this is the point at which humans and mosquitoes interact with each other. And this is the removal term that depends upon this quantity zeta. So all of these quantities are defined here. This is for the mosquitoes. This is I dot so this is the increase in the number of infected mosquitoes when a human is bitten by a mosquito. And this is the death rate of the mosquito. So A is the mosquito biting rate, B is mosquito to human transmission probability per bite, I is the number of infected mosquitoes. So this is exactly the sort of structure that I would have written down earlier. I mean, it says how many bites are there, what is the probability per bite of transmitting an infection. That really comes into the factors A, B, and I. And this is the last term, which is really the human recovery rate, the rate at which you switch out of the infected compartment. And over here I have A and C, mosquito biting rate times human to mosquito transmission probability per bite. So now it's the other way around. It's now the mosquito acquiring an infection from a human being. These are the terms that multiply this on the right hand side. And the assumptions that I described earlier are listed here. And delta is the death rate, the mosquito death rate, all of which you can assume. So the mosquito biting assumption leads to an asymmetry in the transmission term. Remember that in the SIR model, these two terms were exactly the negative of each other. But here there are different terms that for mosquitoes biting humans and humans being bitten by mosquitoes here in terms of who's transferring the infection to whom. So this model was written down for malaria, but pretty much any mosquito-borne infection must have all of these details in. It's the detail, the real sort of complexity of the microbiology comes in the description of what happens at different stages in the mosquito and different stages in the human. But at the level of equations like this, those terms don't matter. It's just, they're all subsumed into these factors, A, B, zeta, etc., etc. So what goes into the model is that the number of mosquitoes is constant, susceptible to infection, blah, blah, blah. These are the quantities that we wrote down over here. And these are the points to remember in this particular case. And from these equations, you can calculate what we did earlier, which is the basic reproductive number. And that turns out to be the product of two reproductive numbers, one for the mosquitoes and one for the human. So this is R0 for the vector to human and R0 for the human to the vector. And this involves, if you put all of these terms together, it's the number of mosquitoes per person. The quantity A is the mosquito biting rate. So biting rate affects you in two ways in both of these terms. It's mosquito biting humans and bites leading to infections in the mosquito. So that can be pulled, you can pull out all of these terms here in terms of these definitions at this point. The mosquito lifespan, the biting rate, etc. All of these enter here and give you these different values of R0. So once you stare at this equation long enough, the definition of R0, you can see that one way of reducing R0 is shorten the duration of infection in the humans. If you can shorten the mosquito lifespan, that also enters here, that is a DM is the mosquito lifespan. That's another way of reducing R0. If you reduce the numbers of mosquitoes, that affects the term M here. If you reduce the biting rate, that affects these other terms, the A term that sits over there. So these, as we argued with the earlier definition of R0, if you understand what R0 is in terms of some underlying model for how bites happen, how efficient are bites in transmitting infection, etc., that will point to the chinks in the armor of this whole cycle of transmission between mosquitoes and humans. And this will, these are ways, all of which have been used in sort of various ways in the discussion of, for example, you can reduce the biting rates by having insecticide impregnated nets. You can reduce the number of mosquitoes by targetedly spraying DDT in areas where mosquitoes are known to breed. All of these other, there are pharmaceutical interventions that could affect this particular quantity here. Just some general points that the well-mixed approximation that goes into these equations doesn't work very well necessarily, partly because mosquito breeding grounds are not well-mixed. They are in specific areas. Mosquito behavior and biology enters much of these quantities. They are not described explicitly here, but more refined models must certainly account for them. Incubation periods, multiple strains, etc. These are all quantities that are important when we think about how malaria is transmitted between individuals. This is a somewhat more complicated model due to Anderson and May, which sort of looks very much like the other model, but there are these various factors that enter here. Again, I don't want to spend time on this, but here is a sort of summary figure of all of the different types of models that people have written down. So the original Ross model had susceptible humans, infected humans, susceptible humans. So it was an SIS model. It had susceptible mosquitoes and infected mosquitoes. Then this was improved by Macdonald, who put in an exposed term over here to account for the latent period within the mosquitoes. Anderson May put in an exposed term for the humans as well, made that more complicated. And then there are a whole bunch of other terms that concentrate on different aspects of migration leading to infection, to increases in infections, the role of environment and weather, socioeconomic factors that lead to some fractions of the population being more affected than others, pre-existing immunity as a function of age, which is detected in this paper here, combinations of all of these effects by Chitnis and Guanshu, etc., all of which look at effects of age in the contracting of infection, effects of prior immunity, immunity gained through multiple bites, how that changes. So you can see that again, as we did with SIRS, EIRS, with the carrier state, etc., when we illustrated this hierarchy of models that got more and more complicated by just adding features that you felt ought to be represented in the way the cycle of infection, the way infection proceeded. The same thing has been done for malaria. There are now many malaria models on the market, and these are important because big organizations like the World Health Organization use these to try and predict what's going to happen over the next 6 months. Where should we target our best efforts? Where should we put in the money into this? Which of these quantities that go up into making R0 for the humans and R0 for the mosquitoes? Which of these are the most sensitive to the interventions that we can try and make? Okay, so that's what I wanted to do today. So I thought I would tell you a little bit about epidemiology and move a little bit away from the modeling, but finally wind up back again with the models. And there will now be a break in my classes for a while. And who among you is going to credit this course? Needs to credit this course? 1, 2, 3. And Janani, where Janani? Okay. So 4 of you, and I suspect, I don't know about Chandrasekhar, but okay. So what I think is I've already given you enough material to understand what is a sort of standard epidemiological model. What I have not done is networks and some other spatial issues related to agent-based models and so on. That would be the material of one more lecture. And then after that, it would be essentially going into any one of these topics in somewhat more detail. But I think I've been fairly fast in, I would normally have stretched this out over more lectures, but the constraints of having to do it a little faster to try and condense it more has been fairly strong.